Science is supposed to be an impartial endeavor. Scientists aren't supposed to get overly attached or emotional about their ideas or theories or hypotheses because someday you might design a test that proves your theory to be wrong, and it's healthiest and best science to be able to just discard it. Since I'm more of a materialist, it seems like there should be some sort of biological or physical basis for things we experience, like religious experiences. The work of Michael Persinger, colloquially called the God helmet, sort of tries to provide a physical explanation for religious experiences, so it should be right up my alley, right? Well, the thing is, the research isn't exactly the best, and that kind of sucks. So, let's talk about why I, and at least one physicist I know, aren't the biggest fans of this work. P.S. I hate doing physics. Welcome back to the channel. Today, we are finally covering a topic I've had in the queue and patron updates for a little bit. When I say topic, I should really say paper, because we're going to go through the original-ish paper for the God helmet. And when I say original-ish, I mean that this is the paper that tends to get pointed to for the God helmet, but a lot of Persinger's work revolved around this sort of topic. I should also point out that the moniker God helmet wasn't the researcher's doing. It was the press talking about his findings. Speaking of, Persinger appeared on Richard Dawkins' BBC program, God on the Brain, where Dawkins had a go in the apparatus, but alas, he felt as though he was in total darkness with a helmet on my head and pleasantly relaxed. In this video, we'll first go through the paper itself without any commentary or critique, although there will be occasional asides to talk about how psych research is usually done or presented. After the paper summary, we will go through my commentary and critique of the paper, and I've even arranged a special guest to help out with part of it. He's got a physics degree and he's married to me, so he has no choice. His input is critical to the critique. Before starting the paper discussion, some quick channel business. Like, if you enjoyed the video, subscribe for more experimental-ish psych research. Comment with your own take on what this research means, or if you have any questions I can help you with. You can also catch me on Twitter. You can join the growing conversation on Discord, or check out my Patreon. We have behind-the-scenes videos, cat videos, monthly live streams, so join today! Alright, on with the helmeting. Starting with an aside. Ooh, so on brand for me. Talking about how the scientific method is typically taught, pretty much up through grad school, versus how it's actually done, is a topic for another day. But I can talk about how we present that research. So, you've got your experiment done and now need to communicate it to the rest of the people who kinda care. Just being realistic here. Psychologist standard for writing and formatting is the American Psychological Association, aka APA, not to be confused with the American Psychiatric Association, a la the DSM, also confusingly known as the APA. Plus there's the American Physics Association, also called the APA, or probably any of the other P fields. Where was I? Right, APA. Psychologists typically use APA format. It's a little bit janky in places, but it's a standard. The flow of a paper in APA format will be intro, method, results, discussion. There's also the abstract, which is the blurb about the paper. Typically, this will be the last thing written, but the first thing read. Typically, this will be a condensed mini version of the paper. This, by all means, is not unique to APA format, but it's not the only ordering of things out there. In the intro, we set up the logic behind the rest of the experiment. So, to start with an analogy, I've heard this described as like an hourglass. So at the start, you'll be pretty broad and eventually get more and more specific until you are talking exactly about your experiment. And how broad you go is determined by the journal you're submitting this to. Something like Nature or Science is for scientists of all types, and so you need to lay that basic groundwork for everybody to understand it. There's more and more specialized journals like memory, where you're only really reaching other memory researchers, so you can start at a somewhat more narrow focus. In all cases, you need to adequately back up your claims with references. This is what I constantly ding Peterson for, inadequate support for his arguments. Towards the end of the intro, you should explicitly lay out what your research statement or question is, followed by your hypothesis and predictions. The research question is the why of the whole thing. It will typically have something about effect of or impact on stuff. The hypothesis and predictions should be a natural continuance of the arguments you've included previously. Next is the methods section. This will include information about who was in your experiment, what materials or equipment you used, and basically what it was like for a participant to go through your experiment in enough detail for someone else to follow your directions and replicate what you did. After the methods are the results. This is where you present your data analysis. Ideally, this is organized to answer the question in a clear and understandable way. Typically, you'll describe the data before diving in with the analytic stuff, or at least at some point in there. It's also okay to relate back to the predictions here. And now it's time to discuss the findings and tie it all back together in the discussion. Going back to the hourglass, we start specific with what this experiment found exactly before working back out to the broader implications. All right, so hopefully now you know what goes where in a psychology paper, so on to the God helmet. The authors indicate their interest in the hippocampus and amygdala brain structures and the relationship to mystical or paranormal experiences. They argue that directly stimulating these structures results in sensations or possibly perceptions not tied to what's really happening. These areas can also be stimulated naturally during a seizure. They further argue that these temporal lobe signs are the focal neurological signs that indicate that something fucky is going on within the central nervous system, specifically the temporal lobe. Temporal lobe signs, given the brain area involved, typically involve some sort of auditory or memory effect. These can include problems hearing various things not tied to physical damage to the hearing bits, so structures in your ear, problems with memory, and hallucinations. Also, since it's not discussed here, the amygdala and hippocampus are in, towards the center of your head, from the temporal lobe. These two structures do a fair bit. The amygdala helps in memory processing, decision-making, and emotional responses. The hippocampus helps out with memory, including spatial memory and navigation. The temporal lobe is responsible for things like memory, sound processing, and language recognition. The occipital lobe is vision stuff. They further argue that these temporal lobe signs should be observable in people who aren't currently epileptic nor having their brain electrically stimulated, and then go on to say that they have found these signs. Furthermore, they indicate that they've been working to infer the functional organization of the temporal lobe, and they've been doing this not just with any experiences, but with near-death or other mystical experiences. They say that vivid experiences, often very personally significant to the experient, have been generated by putting the experience head in a weak magnetic field. In information that should have gone in the method, the authors describe the setup of the room that participants were run in. It is described as an acoustic chamber that participants sat in with nothing distinct on the walls and was illuminated, at least when the experiment was running, by a black and white photography light, so the red light. 3 features are identified as predictive of the experience's likelihood and intensity. First are demand characteristics. This means that the experimenter is either intentionally or unintentionally getting up in the experiment or participant's business. So an example of this would be an experimenter directly interacting with a participant, and whenever they're doing something that supports the hypothesis, the experimenter is enthusiastic and very engaged and, yes, this is good, we're positive. And if the participant starts doing something that's not supportive of the hypothesis, then the experimenter gets kind of withdrawn, negative, not engaged, and participants really pick up on this, and not just humans. In this paper's context, it could be the experimenter dropping hints about what's expected of the participant, results-wise, or religious iconography hanging on the walls, which would lead to a more likely having a religious experience. It could also be a general reputation of the professor influencing things, or just chatter between the students who are the participants and them picking up on what the experiment's about and responding accordingly. Second, personality and cognitive style of the participants. They say that temporal lobe signs are associated with more experiences. The Myers-Briggs Type Indicator, also called the MBTI, also sometimes just called the Myers-Briggs, is included to differentiate between different personality types, and that those who are more feeling, perceiving, or intuitive are more likely to have experiences. We'll talk more about the MBTI later. They also remark that for people who compartmentalize their beliefs from their knowledge, whatever that means, are more likely to believe the experience is caused by the setting. Additionally, this division is more likely to be found in physical science students. Third, don't know. It's not in the paper, whatever it is. Because they can't directly and precisely stimulate the target brain regions, the authors describe how they will use the frequency of magnetic pulses at a set strength to target those regions. Their hypothesis is that these weak magnetic fields, presented at the different frequencies, will alter the normal discharge pattern of neuronal ensembles within deep limbic structures. In plainer English, they predicted that their weak pulsating magnetic field would alter brain function in the hippocampus and amygdala. And because potentially messing around with somebody's brain is kind of serious business, they screened participants for a history of epilepsy and then only ran them in a single treatment condition. 36 undergrad students were assigned to one of 4 treatment conditions in a serial order: 9 Hz, sham field, which is basically just the control, 4 Hz, and 16 Hz. And in case you're not familiar, Hertz is however many per second. So 4 Hertz will be 4 times per second. As a note, 36 participants divided into 4 treatment groups means that there are 9 participants per group. The magnetic field generator was mounted in a set of headphones. There were nails on both sides of the head, each wound 1,040 times with 22 gauge wire. The signal generator was set to generate the field in sharp, spiky increments. For example, the 4 Hz condition would see 4 positive and negative pulses per second. So positive, negative, positive, negative, positive, negative, positive, negative, every second. The room this experiment took place in was in an acoustic chamber without real description of what made it an acoustic chamber. The equipment that wasn't attached to the participant in some way was kept outside the room. The authors say that they had a power frequency field meter and described the magnetic field levels by the nail things than where the person's head would go. They also had some type of additional solenoid attached near the magnetic field generating solenoids to ensure that the fields were being generated. More on solenoids later. In the chamber, near where the participant would be sitting, the background field strength between 1 Hz and 1 kHz was 0.025 milligauss, but varied along one wall due to electrical wiring. They also said that the room was opaque to AM radio frequencies because they couldn't pick up anything on their handheld radio. In a normal setting prior to the treatment day, participants were given the Persinger Personal Philosophy Inventory and the MBTI. Participants were told that it was an experiment on relaxation and that they needed to say anything that came to mind. They were told to sit in the chair and were wired with a number of EEG electrodes and a microphone. During the experiment, participants wore ping-pong ball goggles to block out visual information and listened to a particular 20-minute tape. I'll include a link to what I can find on YouTube that's equivalent, if not a clip of what I found outright, depending on what Algie's whims are today. For 30 minutes, in 5-minute intervals, the magnetic field was turned off and then on. The control group went through the same thing, and the field was still generated, but the plug that took the signal from the generator to the apparatus was unplugged. Everything the person said was recorded onto a tape, and vocalizations were marked on the EEG recording. Once the 30 minutes were up, the participant was gently taken out of that experimental setup with the goggles and the headset and stuff, given the subjective experience questionnaire, and asked to rate the pleasantness of the experience from -2 to +2. Transcripts were made of the participants' tapes. Responses were split into field on or off, or the equivalent time periods for the control group, and were entered into the Whistle Emotions Dictionary to get their arousal and pleasantness scores, as determined by the scorers. Then, these scores for the different words were averaged for field off or on. For clarity, a dictionary such as this one would be empirically derived. What that means is, in one experiment, a bunch of experiments, somehow, you get ratings for a bunch of different words by participants. How pleasant is this word? How arousing is this word? How emotional is this word? What type of emotion? And you average up all the scores for these different words and stick it in a dictionary. And then other researchers can refer to this dictionary as sort of a standardized look at what the valence or emotionality of these different words are. So in this experiment, you have some experimenters, I refer to them as scorers throughout this video, who sit down, look at the transcripts from the participants, and pick out the not filler words, and then go to this dictionary and figure out their arousal and pleasantness. Measures: number of words spoken in a 5-minute period, the previously mentioned arousal and pleasantness scores, average transcript rating, which I'm not entirely sure what that is, the Subjective Experience Questionnaire, or SEQ, score in the 5 clusters, the Complex Partial Epileptic Signs and Control scores, as % yes, from the Persinger PPI test, and the MBTI dimensions, extroversion to introversion, sensing to intuition, thinking to feeling, and judging to perceiving, were derived. I'll explain each, but the EEG data gathered was the alpha rhythm seconds, spikes, and discrete high-frequency bursts. So your brain is electrically active, basically, and we can record this in the EEG data. The character and frequency of that oscillation can indicate what activity level your brain is at. Long, slow waves indicate relaxation and is known as an alpha wave. Higher frequency waves, possibly accompanied by more erratic spikiness, is associated with cognitive processing and is called a gamma wave. So the authors have gone through the EEG data and broken it down into the categories, with presumably some chunks not meeting the criteria for any category and being excluded. Alpha rhythm seconds are 1-second chunks of alpha wave activity. Spikes are sudden bursts that didn't last long, and discrete high-frequency bursts are like spikes but last at least 5 seconds. Basically, these are gamma wave activity. Inter-rater reliability for the experimenters and the participants was verified. So with the experimenters, you want to make sure that the people who are scoring things are consistent with each other. You don't want one person who's really negative and one person who's really positive on everything. You want to make sure they're consistent. They also look to make sure, or to see, if the participants were consistent with themselves. So if they were saying really positive things while they were in the chamber and then came out and were saying really negative things, they wouldn't really be consistent with themselves. A complaining comment that I'll allow myself in summarizing this. Basically, I'm kind of aggravated that the analyses that they set up as they will do in the methods section don't really line up with the analyses that they actually did in the results section. For simplicity and my sanity's sake, I'm going to be sticking with the results that they set up in the methods section. Another grumble: interpretation of the results is not provided as we go through, although this could be a journal format issue, I can't say. So I will be providing some interpretation of what the results mean as we go through them. Final thing before we get into the results. The authors use a lot of non-parametric tests in their analyses. In my Reddit survey analysis video, I talk more about parametric and non-parametric tests, but basically, we want to typically use parametric tests because they're easier, but if you don't meet all the assumptions, you have to use non-parametric tests. And the authors identified that they are using the non-parametric tests to reduce the effect of outliers, which is certainly true. They did a factor analysis on the transcript scores and their subjective experience questionnaire scores. When done well and arguably appropriately, you go into the analysis with the predictors set. You think these predictors are going to account for the variability, and you see how it goes. You can also do this in an exploratory manner, which is sometimes called fishing, or I call the spaghetti method. You basically throw in all of the variables and see what sticks. I was taught that you should only go fishing when you're piloting stuff or messing around with your data. If you are presenting a factor analysis for publication, you should have a model in place beforehand. Well, we play around with data and sometimes things fall out. If you have something interesting fall out, you can share it, but it should be for future research to look into, not the core of your analysis. Table 1 is their descriptive stats here, meaning they describe their data. As you'll see, information about the different ratings between groups is not provided. As such, there's not much to say here other than a lot of people felt dizzy or odd, less so felt good vibrations or heard a ticking sound, and less felt somewhere else, and so on and so forth. In the first factor analysis, looking at transcript scores and subjective experience questionnaire, the factors that fell out as significant were the bodily physical sensation portions of the tests, then the imagination-related scores. Okay, so people who described having some sort of bodily experience had higher subjective equation scores later on. Makes sense. Likewise for imaginings. If you are seeing crazy shit when you're sitting there in that chair, I would imagine that to be reflected in the self-report later on. A separate factor analysis, looking at the MBTI scales and the Persinger test related to temporal lobe signs, had the sensing to intuition and judging to perceiving MBTI axes fall out in the first step, then the control portion of the Persinger test and the extroversion-introversion dimension. So 2 of the MBTI axes fell out as significantly related to the temporal lobe signs. We'll talk about that later on. The second analysis clump broke the data into treatment group, meaning the 3 frequencies or the control, and looked at verbal measures, collapsed across the signals being off and on, interestingly, the transcript scores, Persinger test scores, heart rate, and the 3 EEG measures. They also compared the Persinger test scores with the 4 MBTI axes to see if it impacted the field effects. Again, available descriptive stats for the first part are shown in Table 2. The little A and B indicate significant pairwise comparisons in the ANOVAs. An ANOVA lets us look for differences between groups. It can be done on a single variable between groups or repeating one measure on the same person. Pairwise comparisons are done when you find a difference in a variable and want to know what's driving that significant difference, like which groups are different from each other. There are a ton of comparisons, so to avoid being here all day, we're going to focus on a couple. There was not a significant difference in heart rate. In the temporal lobes, remember the areas of interest here really. There was not a significant difference in the alpha or relaxation activity between treatment groups. There was a reported difference in number of spikes between the control group and one frequency group. 1. The difference was such that the 16 Hz treatment group had more spikes than the control group. But there was also a difference in the gamma or cognitive activity between the control and the same frequency group, with the treatment group having less gamma activity than control. Altogether, here's what I'm getting from this. From the means of the alpha activity, there was a lot of variability between people within conditions that's suggestive that some people were more relaxed than others in all conditions. This will be discussed more later. Table 3 is the participant-provided information broken down by treatment group. If not mentioned here, not a significant difference. There was a main effect or overall difference for the overall subjective experience score, which is not shown directly on the table. There was also a main effect of pleasantness rating, which is shown in the table. The lack of an inclusion of pairwise significance between those main effects indicates that while there was some difference between groups observed, it wasn't enough of a difference to lead to a significant group-by-group difference. There were also main effects of imaginings and depersonalization when the field was on, but pairwise data is again not provided. When the field was off, differences between groups was not found. For now, just note that an interaction between field off and field on is not mentioned. The third analysis clump did a repeated measures comparison for field on versus off and block between the 4 treatment conditions. Repeated measures is a statistical technique where we can buy some extra power by comparing a person's data against themselves. They've been repeatedly measured. So in this experiment, you can compare field on versus field off, exposure 1, exposure 2, exposure 3 for one person, and you get a little extra power that way. But because they collapsed the transcript data across all the blocks, they couldn't do a time comparison there. Okay, so it looks like their MANOVA setup had field status, on or off, block number, 1 through 3, and treatment group as their variable groups. So just as a note, field status and block number would be repeated or within measures because one person saw both or all of those conditions. And the treatment group would be between subjects because each person only saw one of those. For words emitted as the dependent variable, the only significant effect was an interaction between block and field setting, with more words happening after the field was switched on the first time. They mentioned that this coincided with a change in the background music's tempo. They also found some significance for the results centered around the field state and types of words used by participants, so the whistle dictionary scoring thing. They note that this was largely driven by differences in on versus off in the control group versus the treatment groups, which did not significantly differ from each other when off or on. The authors argue that their results strongly indicate that certain subjective experiences can be elicited using their magnetic field setup. Their support for this is from mean comparisons that are not included in the paper, should be Table 1. They argue that these findings support the idea that people who show more temporal lobe signs are more susceptible to magnetic fields. Cognitive styles mentioned again, as related to the MBTI results they found. They say that people who are more intuitive, perceptive, and feeling in the Myers-Briggs display more temporal lobe signs. They note that the lack of field dependence for anything was concerning. Another issue was that the control group was the chatty group when the system was on, not the treatment groups. They finally include that they didn't use a double-blind protocol, and that could maybe be a problem. I discussed how single and double-blind experiments work towards the end of my Jung video, but in this context, a double-blind would mean that the experimenter or the scorer wouldn't know what condition the participant was in. And because this wasn't a double-blind experiment, this could have contaminated their data. They also note that temporal lobe sensitivity is correlated with suggestibility. They conclude that specific experiences can sometimes be generated by pulsed external magnetic fields run across the temporal lobe. Furthermore, studying the brain in this way can reveal information about mystical experiences. Okay, so that was the paper. Time for my comments. I have flags through everything we've just gone through in the paper. So first we'll be visiting some general concerns, then mostly working our way chronologically through those flags in the paper. One thing to keep in mind when reading a published piece of work is where that work has been published. This paper was published in the Journal of Bioelectricity, which is now known as the Electromagnetic Biology and Medicine Journal. This journal focuses on the effects of electromagnetic fields on biology. Its impact factor is currently sitting around 1.6. What this roughly translates to is every article that's published in that journal, on average, will be referenced 1.6 times. So, in psychology research especially, we consider an impact factor of 3 good. So 1.6... Something else to consider is who and what this article is referencing. Ideally, you'll see a healthy mix of authors and journals and things in their reference page. So there might be the same names popping up again and again because, that's what we do. We specialize and we do that. So, of course, you're going to see somebody's work evolve over time. But this paper struck me as odd. Of the 32 references in this paper, 20 of them involve Persinger in some direct capacity. This can indicate that author is sort of operating in a vacuum. Not a cute science look. Final thing here is largely, and admittedly, a life is unfair tantrum on my part. I want to know how this got past the reviewers. This is a peer-reviewed journal. There's problems. How is this not torn apart in the reviewers phase and it was published like it is? Final general issue is the staggering failure to replicate outside of Persinger's lab. Gronkovist and colleagues, notably, were able to publish a failure to replicate that kicked off a bit of an academic fight between their labs. I say notably because an issue we have in science, especially psychology, is the difficulty in publishing null results. Meaning in this case, if you can't get the God helmet to work, you won't find a difference, aka a null result, and will have a more difficult time getting it published. While I frequently have talked about the different psych disorders as being conceptualized as existing along a spectrum, where you have normal, non-disordered behavior on one end and the disordered psychological disorder behavior on the other, I'm not sure this applies to something like epilepsy. So unless the students who score high on this temporal lobe signs test of Persinger's are on the way to developing epilepsy, I'm not sure how valid this measure is. Likewise, I'm kind of skeptical that these temporal lobe signs were actually found by Persinger and colleagues. And also, I'm not sure why they focused on the mystical experiences or near-death experiences like they do, other than it sometimes shows up after a person's had a seizure. Sometimes. Discussion of the weak magnetic field and the room shielding from different frequencies and stuff and what that means. All of that's going to wait until I have the physicist on hand. So that'll be towards the end of the video. Demand characteristics. The bane of experimental psychologists everywhere. It's interesting and strange to me that the authors specifically call out demand characteristics as being predictive for the likelihood of an experience. Part of my alternate explanation for the results that were found in this experiment were that these intro-psych, largely students, walked into a neuropsych lab and were either keyed into sort of what was expected of them just through the information provided as part of the experiment. The experimenter accidentally let something leak or slip that they shouldn't have, or students talk, especially about experiments like what's a good one to sign up for, what's fun, what's weird, what's easy. Information gets out. So, in psychology, especially at the undergrad level, there can be cliquishness, some snobbiness, or elitism. I've seen this at several locations, and I've certainly been guilty of it myself. You've got the counseling students, who typically don't take the harder science or math, hell, even the psych classes, than what is asked of them. You've got the fields that will likely end up in industry kind of sticking together like IO or human factors. You've got the kind of experimental branches, like social psych, who are pretty chill with everyone, but not necessarily taken incredibly seriously. Then we get into the quote-unquote harder psych fields, psychophysics, cognitive, and neuropsych. This is where the snobbiness comes into play. More math and more biology tend to happen for these fields, so you get a sort of science is harder, so I'm better mentality happening. The psychophysicists tend to not be very outwardly snobby, but it can still be there. Cognitive, again, this is all from my experience, the uppityness depends on the student. When I started grad school, was insufferable. I had a bio minor from undergrad. At my school, I had to take a year of calc, a year of physics, and a year of chemistry. No one in my cohort did that. So, more science is better. Therefore, little elitist me, I'm better. And if I'm not careful, sometimes I can slip back into that elitist mindset. Oh yeah, this statistics thing, you would totally be able to understand it if you'd taken a year of calculus. But, and I say this with all the love in my heart, then we've got the neuropsych people. Depending on where you go to school, you might be slicing up rat brains to look at stuff. You might be handling donated human brains to learn the different brain structures and connections and all of that. So there's a lot of anatomy, physiology, memorization, grossness that the other psych fields don't have. And so that sets those guys apart a bit. And all that is fine and dandy. My graduating advisor was a neuropsych person, and she was an incredibly lovely person and terribly down to earth. But I've seen students, neuropsych students, who idolize and almost worship the neuropsych professors. And it seems to happen more in neuropsychology than the other fields of psychology. Not sure why. I saw it happen where I did my undergrad, I saw it happen where I did my grad, and I saw it happen when I was adjuncting. These professors are the smarty smarts. Their classes are the hard ones. Although, true story, the professor who handled some of the neuropsychology stuff for the grad level, her classes were a joke. It wasn't uncommon for questions to appear on her tests like, on the Alzheimer's slide, what color was the background? And these weren't open book tests. Just why Why do you think that's a good question? No. But back to what I was saying. The general perception among students, especially neuropsych students, is that those professors are the smartest ones. Their classes are the hardest, and that research is the most worthy of doing. I thought maybe this was just a fluke of my undergrad. Got to grad school and nope, still happening there. Maybe it's a United States thing. Moved to a different country, same damn thing happened, so definitely turning into a trend. And this perceived stratification is known to the intro psych students within a couple weeks, and it's all the worst when the professor sort of feeds into this hero worship. Now, I'm not saying this is what is going on with Persinger in this research. However, Persinger himself is the one who gave me the worst conference presentation experience of my life. And that's on top of Richard, my advisor, disappearing just every poster session. At a little conference, I had a poster of some of my consciousness-related research, and up walks Persinger in his infamous three-piece suit and asks me to go through it. Cool, no problem. That's why I'm here. I start by saying that I'm interested in consciousness and start to get ready to go through the spiel for the poster, and he stops me. He stops me and says, "Oh, consciousness? Yeah, we solved that problem in my lab, but do go on." I wanted to deck him and I wanted to run away. Both were not options at a poster presentation, so muster up whatever I can and finish the presentation. He thanks me for my time and moves on. I guess maybe I'm lucky that's the worst, but still, it's the worst experience I've had. In doing research for the Reddit religious survey video I did a while ago, I found out that the God helmet was Persinger's doing, and my reaction was, of fucking course it was. So Persinger seemed like the type of guy who would actively cultivate this cult of personality, which the neuropsych students would be drawn to, magnetically. And this could translate into demand characteristics, especially for the more suggestible participants, but more on that in a second. Persinger's reaction to Dawkins not experiencing anything really, being that of course he didn't because he was low on the temporal lobe sign score, made me think of something. The personal philosophy inventory that Persinger and colleagues developed sounds like it could rather function as an index for how suggestible someone is, and not what they say it's measuring. The Myers-Briggs Type Indicator, or MBTI, is a personality test that is based off of the work by one Carl Jung. That Jung. So if this is the first video of mine you're seeing, hi, welcome. I don't think that Jung's work holds up to scientific scrutiny. And so a personality test based on that work, this is not good. By the way, I did a video on Jung, feel free to check that out, link in description. Worryingly, the reliability and validity of the MBTI has been questioned. So reliability in this context means that you will get consistent personality scores over time, and that's not what happens. I mean, some people can test pretty consistently, but others will have drastic personality shifts between test takings, so that's not good. And validity means that we're measuring what we say we're measuring, so in this case, personality. But the constructs that the MBTI is measuring are based on the Jungian constructs or archetypes, and those aren't exactly valid. So the MBTI as a personality test probably isn't that valid. So yeah, instead of using something like the Big Five model of personality that is more reliable and more valid, they decided to go with the Myers-Briggs. Neat. Although their discussion of cognitive style fits right on in with this. Throwaway comment about belief compartmentalization. What? If this is such a big thing, why wasn't this one of the demographic measures that they took so that they could measure this? And how do they know this division is occurring? There is no reference attached to this statement, so it sounds like an untested observation. Also, just a general, what the fuck? If I went in for a God helmet session, and if I actually managed to have an experience, I would ascribe it to whatever I was being put through and not some religious experience. So, are my beliefs compartmentalized? Is this similar to Peterson and his no true atheists? Just, I can't. Not even. The lameness of the hypothesis. Part of why I was foreshadowing like crazy in the paper discussion was to illustrate a well-written paper versus one that isn't. A strong hypothesis won't be buried in text and make no real predictions. Oh, it's going to alter brain function somehow. Brilliant. The serial order assignment of participants and no additional information. Typically, when doing a scientific experiment, especially in psychology, we are mindful of time of day or other weird order effects. So practically, a time of day effect means that if you're assigning people in serial order and you've got, let's say, 3 conditions, one condition will always be in the morning, one condition will always be right before lunchtime, and the other condition will be at the end of the day, just with your lab schedule or whatever. And so if you get a significant difference between those groups, you can't say that it was the manipulation you did because you have a time of day effect. In the morning, you've got participants who are bright-eyed and bushy-tailed, or possibly hungover, either way. Right before lunchtime, they're probably hangry, and at the end of the day, they're probably tired and ready to not be at school anymore. And that's a time of day effect, and that's why we don't do this where we just put people in conditions in order. We randomize it. And then we check to make sure that our randomization of what condition people are being assigned to doesn't somehow accidentally end up with that time of day effect. Comment about sample size. 36 participants can be too few, plenty, or even too much, depending on the type of experiment. We typically determine sample size based on some napkin math. Basically, how big of an effect size or difference between groups are you expecting? And from that, how many participants you need per group is spit out through some math. Some experimenters do kind of questionable things where they have a teeny tiny effect size that arguably doesn't really have real-world significance, but they throw hundreds of people at it and get significant results, and then build an entire career out of this itty-bitty effect and somehow get an NSF grant. Anyways, given the abundance of references on this type of stuff from their own lab, they probably had a guesstimate of what their effect size was and how many participants they'd need. However, it would seem like they didn't have enough. The huge variability in data, which we'll talk about more in a second, could have been mitigated by a larger sample size, if this was an actual effect. If this wasn't an actual effect, then adding more participants wouldn't really do much to help with the noise. The room this experiment was run in. As previously mentioned, there were neuropsych people at my grad school, and some of them did EEG research. And when I started, they had nice new shiny lab spaces built, and prior to that, what was actually my office was used as an EEG space. In both cases, the experimental space where the participant would sit was surrounded by a Faraday cage. We'll talk about this more when we have the physicist, but Faraday cages basically shield from electromagnetic interference by grounding out the interference to the cage. The less nice version that was previously housed in my office had everything that the participant needed, basically, surrounded by a box of chicken wire. The nice version had the shielding built into the walls. There's plenty of repetition in this paper that the room was acoustically shielded, but nothing about electromagnetic shielding, and that could have fucked with their EEG readings. Let's talk a little about visual sensory deprivation. Basically, when you wear ping pong goggles, you are experiencing visual sensory deprivation, assuming you've got uniform light. You can also do this with pure pitch black, but that's harder to accomplish in a lab setting. Sensory deprivation, even if you're just blocking vision, leads to unregulated cortical firing for that sense. This can result in hallucinations, seeing vague shapes, or solid objects, or even deceased loved ones. Adding in a trippy sound experience and the feelings of oddness or strange sensations, seeing vivid imagery, or hearing things seems to have an explanation that has nothing to do with the weak magnetic field. To me, the demand characteristics and the sensory deprivation are really what's driving the effects that they found, not the weak magnetic field. We tend to want to make sure the manipulation we're doing in the experiment is the only thing differing between groups. A concern with running everyone in the off to on pattern is this ordering could have done something screwy with the data. Typically, what would be done in this sort of case is to counterbalance the exposure order. Half of the participants get the off first, half of the participants get the on first. Then, at the very end when you're analyzing your data, you compare the measure between those two groups to see if exposure order did or did not matter. But given their small sample size, I imagine they didn't do this because splitting 9 into 2 is even smaller than what they've already got. The background music. So, the magnetic stuff went on for 30 minutes. This musical tape was 20 minutes long. What did they do? Did they just let it stop and then leave silence for 10 minutes? Did they have somebody sneak in and flip the tape around? Did they have a player that would automatically just switch to the other side of the tape? This information is not included, but it's a glaring issue. Let's talk about the variability in their data. I touched on this a little bit ago with the sample size, but their errors are huge. When the standard deviation is as big or bigger than your mean, you did something wrong somewhere. This could have been helped probably by trimming outliers or running some more participants. So, in excluding outliers, you need to make sure you're doing this right. You don't want to just start trimming participant data who don't fit with your hypothesis. That's terrible science. Terrible. But if you have notes about what participants are doing or things that are going on, like if they have a breakdown mid-experiment, then you have just cause to exclude them. Likewise, if you are just shy of having a significant effect, you might need to reconsider your effect size. Maybe it wasn't as strong as you thought. Check the research, check what other people do, see what numbers they use, and possibly run a couple more participants. I had to do that a couple times, and it's sort of the praxis of doing experimental psychology. One of the items from the subjective experience questionnaire was hearing a ticking sound. So something that I've just been continuously curious about is if participants could hear when this thing was on. I'm unclear if it was just completely silent or if it maybe made a little bit of a hum or a buzz or something. Or they were just describing the weird music that was playing. Another statistics-related comment. Let's talk about alpha. Alpha is the likelihood of you getting a significant effect or result or whatever when there actually wasn't one there. So in psychology research, we tend to use a probability of 0.05. What this means is that, let's say you don't have an effect. 5 times out of 100, you'll just get the participants lined up in the right groups where it looks like you have an effect when you don't actually have one. The thing is, though, when you do a bunch of comparisons on the same data or the same stuff, you can't just leave the alpha at 0.05. You have to correct for these repeated testings, and there's a couple ways to do it. I'm partial to Bonferroni myself. Basically, what you're doing is setting the bar higher. Instead of 5 times out of 100, you could expect this through chance alone. Now it's at 1 out of 100, or even less than that. I see no indication that any sort of correction was done in this paper, and they did a bunch of repeated testing. So, as that happens, the likelihood of significance purely by luck increases. Also, not acute science luck. I'm suspicious when I see nearly an entire column of zeros. That none of the control group had transcript ratings despite talking a bunch doesn't add up. This could be related to the demand characteristic stuff I talked about earlier. You don't want to go against the neuropsych professor's way of scoring stuff, or you want to score it in such a way that helps the professor's experiment turn out true. This is also related to the issue of not double-blinding this experiment. The people scoring these tests shouldn't have known what condition participants were in, but it seems like they did, and that's a huge problem. So yeah, I wasn't terribly convinced by this paper, and we haven't even gotten into the questionable physics of it yet. Alright, so for this last chunk of the video, as promised, I have acquired myself a PhD physicist to help illustrate or elucidate or whatever the physicsy parts of this paper, because, like most psychologists, my physics ability is piss poor, and my eyes tend to glaze over on this stuff, and I kind of suspect that's how a lot of the stuff got published, where the information doesn't necessarily make sense to somebody who is in physics, but for psychologists, like, yeah, sounds good, whatever. So I have my husband who has a PhD in physics and done various fun things. Say hi.
Hi.
So he has experience doing all kinds of crazy things.
Lasers. Motherfucking lasers and shit.
Lasers. Knobbing, little fidgety, breaky fiber optic cables to do things, and much, much more. So,
trying to keep it discreet, I guess.
Yeah. A discrete physicist?
Discrete physicist.
So, continuous physicist. Okay, so the first thing is this issue of the weak magnetic field strength used in this experiment versus the Earth's magnetic field and other things that's just surrounding us in our homes or when we go outside. Can you speak to that?
Right, so the Earth's magnetic field is roughly half a Gauss, where Gauss is the unit we just use for magnetic fields in this scale. And it's the unit that they use in this paper as well. So to be consistent, we'll only talk about Gauss, or try to.
What's 1 Gauss?
Again, a Gauss is just a CGS unit.
And what's CGS?
CGS is centimeters, grams, seconds. It's typically the astronomers use CGS. Most of us use SI, where the SI unit for magnetic field is Tesla. So you may have heard of Tesla. It's just a unit scale. Like there is no what would you call the mass?
Standard?
There's no like unit standard that makes sense for a Gauss in my mind, other than the Earth's magnetic field is about half a Gauss. So sort of the natural is order 1 Gauss. You know, like magnets and stuff are all sort of on the Gauss scale typically, unless you're talking about a big ass magnet like an MRI magnet.
Okay.
Just to relate that to the paper, again, they're using half a milligauss roughly fields that they're generating with these little solenoids, and then.
What's a solenoid?
A solenoid is a coil that you run current through to generate a magnetic field, or you can do the opposite. You can measure the current of a solenoid and change the magnetic field around it, and it'll sense that magnetic field and give you a current back. So it's just a coil, an electrical wire coil, essentially, and you can use it to. Most of the time, we think of things like solenoid valves or something where we actually use a current to create motion to open a valve or something like that.
Okay, so back before I distracted you, so the Earth's magnetic field is half a Gauss, and what can you expect around the house?
So you can expect your household appliances to generate on the order of 1 milligauss. So, you know, on the same order or slightly bigger than the experiment, from the 60 hertz alternating current that's delivered to your house that's used to turn motors in blenders, dryers, whatever. So all of this stuff generates magnetic fields in your house, and you're exposed to it constantly at 60 hertz.
Okay, and they were in the 4 to 12 hertz range, right?
Yeah, they generate an interesting pulse shape. They create with a function generator that spits out a sine wave, and then they use a circuit to create an interesting pulse shape. And
blah, blah, blah.
Well, there is one of the problems I have with the paper and the layout is they say that they generate this pulse shape with this circuit, and they don't offer any reason why it would do anything.
Yeah.
It's sort of magical. Just like they all lay it out as, let's just try this, which is, to me, it's fine, but you need to have a pretty damn strong effect to say it worked. Yeah. In that case. If you have no physical mechanism to say why it would work, you should be doing a threshold experiment with a very high threshold for significance, in my opinion. Mm-hmm. And this does not meet that. It just sounds like, let's try this. Oh, look, God. Like, how the fuck did that work? It doesn't. Like, it's very wishy-washy, very wooey. Mm-hmm. It's kind of gross.
Okay, so the next issue that showed up a couple times previously is, to me, it doesn't sound like the room was actually shielded. And this is a huge issue for me because they were using EEG electrodes, and the labs that I've seen that do serious EEG data, they have their person who's being recorded in a Faraday cage. Like, the room is shielded. So first, how do Faraday cages actually work?
All right, so Faraday cages are constructed out of a conductor, typically a metal, that if an external electric field is induces charge on the cage, that cancels the field out on the inside. So it basically nulls a field out on the inside. It doesn't do anything for static magnetic fields, by the way. So Earth's magnetic field is still a factor in this experiment. Their experimenters' brains, or experimentees' brains, are subjected still to Earth's magnetic field through this whole experiment. Mm-hmm. Oh, and, well, whether or not they had a Faraday cage is up for question, right? They say that their room was opaque to AM frequencies, but that's not the same thing as what you would expect from pickup from your AC noise. So they even say on one of the walls they measure noise.
Yeah. So that they had leakage coming in from electrical wiring in one of the walls really indicates that they didn't do any of this shielding that seems to be the norm when you're doing EEG research. So that's another problem.
Yeah. One of the gross things I noticed reading the paper was they explain away this problem by saying that typically if there is crosstalk in the noise from something, it's very large. But they don't say, like, it could be any strength of crosstalk. So the fact that you say that you don't see a huge crosstalk means you have none is bullshit.
Well, and also if they had large signals coming in, wouldn't it sort of mute whatever they're trying to do potentially?
Yeah,
so
what they say is there's only a small effect seen from, say, the EEG during when the pulsing has happened, right? And that's part of their conclusion that this does something. And they say, well, we would have expected a huge effect if they were crosstalk, if there was crosstalk.
Right.
But that's bullshit. It could be any crosstalk strength. Right. It's just gross. It's again another example of something that doesn't pass the smell test.
So one of the conclusions from the paper from these authors was that some people are more susceptible to magnetic field, and the people who have these higher temporal lobe signs as determined by the Persinger personal philosophy inventory. Yeah, so does it seem likely that some people would be more sensitive to magnetic fields than others in a way that meaningfully impacts their consciousness or their cognitive stuff?
It seems extremely unlikely to me. Again, we evolved, adapted to Earth's magnetic field that's 1,000 times larger than these experimental fields. And our natural frequency, say turning our head, is something like 1 hertz. You could probably do a few hertz even. And you don't see God every time you spin in your chair. So I don't see any physical reason why these tiny fields would have any impact to bio/neurological processes. And it just seems like these fields are just too weak to do anything actually in the brain, given we've developed in such a large magnetic field, respectively.
So that segues nicely into talking about like TMS and MRI. I think I've talked about MRI on the channel before. If you're not familiar, that stands for magnetic resonance imaging, and it's used a lot in medicine. You may have experienced it where you go into a tube and it clangs for a while. That's an MRI. And so in that, how do they work? Let's start there.
Oh, how do they work? This is quite a detailed question. It sets up 2 magnetic fields, actually. One is a gradient along your body from head to toe, and another is just another gradient, probably perpendicular to that. And the whole idea is that depending on the field, which is why they set the gradient up head to toe, and the other gradient, they induce another field that then resonates with hydrogen atoms in your body, which is from water, essentially, right? Hydrogen is H2O. So the hydrogen is just a proton, and it has a spin. And so they're actually flipping the spin of these hydrogen atoms, and the frequency that they do it it depends on where it is in your body based on the fact that they set up these gradients, and they can image basically the water density in your body that way.
Yeah, and so like the water composition of your body isn't uniform. Some things have more water, some things have less water. So when you get into functional MRI, their fMRI, which we use for experiments, looking at changes in the brain, they are looking at the BOLD signal, which stands for the blood oxygen level D. I forget what the D is, but basically they're looking for changes in blood flow, and that's thought to relate to either increases or decreases in the neuron's activity level.
I'm not an expert, and I don't know, but my understanding of functional MRI is just that it's they have a time resolution. So it's a bigger magnet, and essentially that means that they can do the imaging fast enough that they can actually image blood flow instead of just water content per millimeter.
To get absolutely out into the weeds here.
Yeah.
So when you go in for an fMRI experiment, first they'll do what's called an anatomical scan, where they are doing it slow, steady, getting all of the nitty-gritty detail of your brain. That gives you good spatial resolution, but poor temporal resolution because it can take like an hour. So then when you're doing the actual experiment itself, you get good temporal frequency, so you can see the fast changes in the blood activity, but you lose some of that spatial resolution. So they have to use the anatomical scan to figure out where the different parts in your brain are, and then move from there.
They map them onto the...
Yeah, kind of. That's fMRI and MRI. So then TMS stands for transcranial magnetic stimulation, and that is something that kept popping up in my head with this paper because there is a technique, TMS and repetitive TMS or RTMS, which is being used for like medical applications or tested at least. So these, again, it's very, well, not very, kind of similar where you stick a magnet near somebody's head, and you can talk about the difference between the TMS and the God helmet better than I can probably.
Yeah, so what we didn't talk about was the scale of the magnets used in these instruments. And so again, Earth's magnetic field is half a Gauss, and these are 60,000 times larger magnetic fields than Earth's magnetic field. So 60 million times larger than the God helmet's magnetic field. And these actually do really affect, the physics of matter in your brain, or the in the case of TMS, what it does is it induces a current, an actual current in your brain that can change the action potential of neurons, and that is sort of how it functions. I'm not going to say anything about how action potentials work, but...
So quick, very dirty version. Your brain has a bunch of neurons that are like the cells that make up your brain and help you with the thinking and the remembering and the stuff. So when we talk about a neuron firing and action potentials, that's how a neuron transmits information to whatever neuron it's connected to. And this is an bioelectrical process. So there's a whole bit about like sodium and potassium and blah, blah, blah. But basically, in order for an action potential to happen, a certain electrical threshold has to be reached. And once that threshold's reached, it is a ballistic process. The action potential happens, and then the cell has to recover. And so TMS, so depending on the frequency of the magnetic field that's being used in the TMS, you can either inhibit cortical firing, where it makes it harder for the neurons to send those signals, or you can provoke cortical firing, where you're sort of cheating the system and making action potentials happen by inducing that necessary electrical activity in that neuron.
Right. So you can either inhibit or induce firing of neurons. And the beauty with TMS is you can shape the magnetic fields so that you can to some degree pinpoint areas of the brain that you do this to. So you can do things to motor or other areas of your brain, etc.
Yeah. You researched TMS a little bit today to help with this. And basically, I think you found something about TMS only really working for the cortex, right?
Yeah, so it seems to... It has to be somewhere where there's enough, I guess, resistance in the brain matter it would probably be the opposite, to create a current. It basically has to be a part of the brain that you can induce a current in, and it tends to be in the white matter and not in the gray matter.
Right. So in the cortical areas, so like we saw a demonstration of this when we were in grad school, where the TMS thing was held up against the volunteer's head, and it sounded a lot cooler, but it was still kind of cool. They were able to induce a muscle movement in the volunteer's finger that was rhythmic in time with the TMS unit. It wasn't a voluntary movement for him. They were directly activating that motor center in his brain to cause that movement to happen. As it was explained to us in that demonstration, as far as we know, this is a safe thing because we're only hitting the cortical areas. If it was able to interfere with activity in like the brainstem, that would be bad news. And you could potentially kill somebody if that was the case. But it doesn't, because it doesn't have the right
electrical properties essentially.
Yeah, so as far as we understand, TMS is safe, although there's always that caveat when you're filling out the informed consent forms that we don't know what's going to happen 50 or 60 years down the road. Yeah. So as far as we know, it's safe, but there is that possibility that there might be some long-term thing. Right. And it does seem to be able to change function or cognition stuff on different timescales. So some experiments have knocked out vision for participants using the TMS for, I think, something like 10 or 15 minutes. They just inhibit activity in the occipital lobe, or the visual cortex, and people can't see. They're functionally blind. But it goes away, it gets better. On a longer term timescale, I know that there's work on using TMS to help out with things like depression, OCD, other things where you try to change the wiring of the brain, at least in the short term, to help the person with their functioning. And so this is typically used when you are past the drug resistance stage. You might also be considering electroconvulsive therapy, which also works by blunt force changing how the electrical properties of your brain's working. So the advantage of TMS is you don't have to induce seizures in people. You just go in, sit through the TMS sessions for like a couple hours, I think, for a week or something, and people have some improvement in their depressive symptoms. It doesn't seem to last super long term, so these treatments do have to be repeated though. I know it's thought to work through long-term potentiation and long-term depression, but that is super out in the weeds, so we are not going to go there.
Fair enough.
So do remember, dear viewers, that this paper originally said that they were trying to affect the amygdala and the hippocampus activity. Those are deep brain regions that are not part of the cortex. So it doesn't seem, at least with TMS, like we'd be able to impact those. I don't know why a weaker signal would be able to.
Right. Well, they offer absolutely no physical mechanisms that this would work.
Okay. So, conclusion of the physicist portion, what is your opinion on the whole of the paper?
So given the weak fields, especially compared to natural conditions and the sort of not spectacular frequencies that are sort of within the realm of biological motor movements, I find the fact that this could do anything quite hard to believe. I suspect that this is due to the fact that they're biased researchers in terms of interpreting their results. The results are highly subjective, it seems like, in terms of their looking at what the subjects say and interpreting that in terms of what they think. They don't offer any good mechanisms to suggest why their magic circuit and their silly little field would do anything to the brain. I mean, it has no physical justification on how it could potentially activate any brain regions. It doesn't even speculate on it, which is sort of sad. They developed their circuit based on simplicity. I don't know, yeah. There's a lot of funny things about it. I just don't like the magicness of it. It feels wooey, it feels gross.
All right, well, thank you for your time. And let's wrap up this video. So, that's the God helmet. I'm not terribly impressed or convinced by it, but that's me. Tell me what you think in the comments, and until next time, bye!